Okay, let's talk about how we grow mushroom in our lab. First, we need a fridge in order to store all our sample, our culture, and also our mushroom spawn. So mushroom is a fungi, so it starts with this stuff. This is a mycelium of oyster mushroom. We have the dried oyster mushroom there. So normally when you go to the supermarket, you find a mushroom, or you walk through the park and you find a mushroom, what you see there is just a fruit of fungi. So the body of this stuff is basically the mycelium. So this mycelium is basically a collection of tiny hyphae, or basically tiny branch network of fibers lah. So this is a mycelium of grey oyster mushroom. So normally in order to get this, what you can do is that you can cut the fresh mushroom, and then you put it on the on the petri dish, on the agar, and over time it will grow and like this mycelium lah. So that's what we call cloning. The other way to get this thing is that wait the mushroom to mature until it release a spore here. So the mushroom will release the spore through the gill. So this is oyster mushroom. They release the spore, white spore, through this gill. Take that spore and then sprinkle on this agar, and then you will get over time the mycelium will grow lah. So that is basically breeding. Okay, so there are basically two ways in order to get this mycelium, either through cloning or through breeding. And when you get this mycelium grown like this, you can cut part of it and put it on something like is nutritionist. Okay, so this is example of mushroom spawn. Mushroom spawn is basically simply a mycelium on a nutritionist grain. In this case, we use what we call the sorghum there. So we take a part of this stuff, mycelium, and then throw it inside the this plastic bag that have sort of like pre-soaked and sterile grain, and over time it will take over the grain, and you get this stuff lah, the mass of mycelium on a grain. So when you have this thing, we call this as a spawn. In Malay we call it benih lah. So we can use this spawn to inoculate our substrate, because mushroom need food, because mushroom is not a plant. Plant can make their own food via photosynthesis. That mushroom, they need to obtain their own food. They need to source their own food lah, like us human. So we have this spawn. What we did is that in order to grow mushroom, we crush this spawn like this, crush this spawn, and then put in our sterilized substrate. So this example of substrate, you can use any lignocellulosic material like sawdust or whatever, and then you sterilize this first, and then only then you take this and then put inside this, and then it will sort of colonize the entire substrate. And then once it's done colonizing, once this thing turn like whitish like that, you can put this inside the fruiting chamber. Okay, and then you get the mushroom lah. Okay, so we have this oyster mushroom here. We also work with also Ganoderma. So this is Gano, Lingzi, in other words, they call also like a Lingzi mushroom, the hard bracket polypore mushroom. So this example of what we call Ganoderma spawn. Okay, so what else we have here? So yeah, this fridge is important for us to preserve our strain, and also to sort of like hibernate our spawn from getting too matured lah. So when we incubate our spawn, and then we don't want to use it straight away, we just put inside this fridge so that we can use it later lah. Normally we store it for like 3 months or 6 months here. We still can see the mushroom can coming out from this spawn lah. So we have oyster, we have the Gano. So up to now we just work with the oyster mushroom, grey oyster mushroom, and also Ganoderma. Maybe in the future we can expand our strain or our mushroom type. But this is the first important thing that you need to have in order to grow mushroom in the lab, the fridge. This is our incubation chamber. So we have two of those. So let's see this chamber first. Incubation chamber is simply a place where we put our substrate that's already inoculated by the mycelium to grow. So this is oyster mushroom on some substrate. So we use this plastic bag here. So we seal the plastic bag, and then this plastic bag have some filter here. This is 0.2 micron filter. So the purpose of this filter is that to allow the air exchange. Because the mycelium need to breathe like us, and also this filter, they prevent the moisture from going out or coming in, so it's retain the moisture of our substrate here, because mycelium needs some moisture to grow, and also it's prevent some contaminant like bacteria to go into lah. So we have this rack in order to put our sample bag, and we use a different type of substrate there. And this is Ganoderma, meaning that we inoculate this stuff with what we call Gano spawn, like we see before. And for this what we call chamber, we regulate our temperature to 28 degrees Celsius. So this is our regulator down here. Okay, so how we regulate it? Just using a small heater fan. It's small but it's effective because we only have a very small chamber. So this chamber, the dimension for this chamber is basically 4 feet by 4 feet by what we call this 8 feet, I think. Okay, so that's the dimension of our chamber. So inside our chamber, apart from this what we call the heater, we also have the fan, rotating fan here, to allow some circulation of air lah. And also we have this exhaust fan out there, so in order to bring in what we call the fresh air in. Okay, so because here we have some opening there, some opening there, some opening there, so the air can come in and then go out there, and it will sort of like replacing the air inside here lah. But for incubation chamber, that's not really critical, because for mycelium to grow, you don't really need low CO2. I mean that mycelium can tolerate high CO2, high carbon dioxide level lah. So it doesn't really matter lah. Actually, if we don't have this also, it doesn't really matter. If we don't have this also, it's still okay. Because mycelium can tolerate that lah. The most important thing normally is the temperature lah. So the temperature that we use here in this chamber is around 28 to 30 Celsius. Because our grey oyster mushroom and also we use Ganoderma here, they thrive at that range lah. And maybe if you use a very exotic mushroom like lion's mane or other shiitake or whatever, maybe they need a different temperature. But for oyster mushroom and this Ganoderma, generally you can get by using like 28, 29, 30 Celsius like that. Okay, so this is our chamber. Okay, so this is our first chamber. So we let our substrate in this bag to grow, and over time it will grow fully like this lah. So this is a fully grown substrate. Okay, this oyster mushroom mycelium take over the entire substrate, and then when you get this whitish, fully whitish what we call bag like this, you can transfer it into the fruiting chamber. Okay, fruiting chamber is different. This is incubation chamber. Okay, so let's see. So how we place our what we call our sample inside this chamber. So this side. At least you get the idea lah. Okay, so that's the thing. Okay, so this actually can be closed lah, because they have a door here. Okay, they have a door here. For mushroom, for mycelium, you don't need any light lah. You don't need any light, so that's why there are no lighting, no whatever. Even if we have no what I call no fan, also it's okay. The important thing just some exchange of air lah. I mean replace this air inside this. Okay, so that's it for this chamber. So let's turn into this chamber. Okay, so in Asia normally they grow mushroom by this block lah, this cylindrical block. In what I call in the West, Europe, West, normally they grow mushroom in this filter bag. Okay, it's just a preference. It's the same thing. So you can see there, that is, I can see on the what I call on the back here. So that is mycelium actually. Okay, the mycelium. So the strand of hype. So this the big, if you see something big like this, the mycelium like this, we call that a rhizomorphic mycelium, which is a good healthy mycelium lah. Okay, so that you can see, that's okay, so that is the thing. Okay, let's see closely. Okay, there. Okay, so this is a bag of what we call substrate inoculated by the mycelium, and normally those in the West they prefer this, but in Asia we normally use this cylindrical block. Okay, the way how we put our what we call our spawn before is that we just throw it inside this bag and then we seal the bag. That's it. But for this cylindrical block, you have this cap. You have this cap. So normally what you do is that you open this cap, and then you put the what I call the spawn on the top here. And because this block is normally positioned upward like this, you will see the mycelium running down. Okay, so all the sample is already fully matured. We call it matured lah. When the substrate already colonized by the mycelium, we call it matured substrate lah. So but at the beginning, this thing, imagine this thing like what you call, like brown in color. So you put some spawn inside on the top, you will see the spawn going down until the end lah, until the base. So typically for this size of what we call of the cylindrical block, depending on the strain, for oyster mushroom it will take around anywhere from 1 month to 2 months lah. Typically 2 months lah, from the mycelium from the top goes until the bottom, because we use only a few amount of spawn on the top here. Okay, compared to this, normally we just throw a lot of spawn. So in maybe like a week or 2 weeks, it's already colonized fully like this. So it's basically how fast the mycelium colonize your substrate depend on the amount lah that you put inside the bag. So here is something else lah. Okay, so this is how we what I call, we put our cylindrical block in our chamber. So the condition is similar like before, 28 Celsius, we maintain 28 Celsius, and also we have a fan for circulation, and also we have the exhaust fan out there to replace the what I call the air once in a while. Okay, so this our another incubation chamber. Okay. Okay. This is our fruiting chamber where we trigger the formation of our mushroom here. So this is a grey oyster mushroom from our fully colonized block that we take from our incubation chamber. This chamber have similar dimension with our incubation chamber before, 4 feet width, 4 feet depth, and also 8 feet height. The difference between this and previous chamber is that we have lighting there, we have humidifier below here, and also we have aircon there. So that's aircon actually is connected to this portable aircon outside, because we don't want putting this aircon inside this chamber because of this humidity. The reason why we need to make all this setup, because in order to trigger the mushroom formation, you need to give this colonized what we call colonized block a certain condition. Typically you need to mimic the condition after the rain, which is cold and also humid. So cold, we use aircon, and humid, we use humidifier. Normally in your fridge it's cold, but also dry. In order to get humid, you need to add this humidifier lah. And also lighting here, Normally fungus doesn't need light in order to grow because they don't make what I call food through photosynthesis. They sort of like find their own food from this lignocellulosic substrate here. So they don't really need a light actually to grow, but light is important in order to trigger the pinning. The pinning is like this lah. This yeah, they need light in order to start to pin lah, the small mushroom to grow out lah. So this is how it's like. We put our what I call two beaker at the bottom here. We fill up with some what I call water in order to let the humidity to preserve the humidity lah. Although we have our humidifier here, the big humidifier here, but we just add some reserve here water so that over time it will evaporate and let this chamber become cold lah. Okay, same like before, we have rack and then we have our what we call our substrate bag. So normally in Asia, when you have this cylindrical substrate, in order to grow the mushroom, you lay it down like this, so that the mushroom coming out like that lah, like that's coming out like that. Okay, so it's pinned first here, and then over time it goes big like this. So you can see there are small mushroom over there. Yeah. Okay, so this is a mushroom also. This thing in the morning, they just small. The mushroom is still small and it's not even curl yet, but then mushroom have very short lifespan, so you need to harvest it as fast as possible lah. I mean before they curl like this. I mean when you see the mushroom start to curl, that's what we call show a sign that the mushroom is ready to release their spore lah. So the curly condition here is basically a condition where the mushroom get ready to release their spore. Okay, so normally if you want to harvest it, maybe you harvest before it's being curled lah. So this yesterday is still okay. I mean it's still not curled and still nice, but today it's already curled like this. So when you see something like this, small like this, you need to take it as soon as that you need to be ready lah. Normally in 24 hours this thing will turn into this. Okay, they have mushroom have very short lifespan, so you need to be aware lah. Okay, so this also you can see they are start coming out, the small mushroom there. Okay, so we also have a gano. We also put gano here. So down here you have gano. So our gano. So this is the mist. Wait. Okay, so this is our gano. Okay, we put there inside this what I call filter bag. We also put in the cylindrical block. So it just grew like that, and we put our gano at the bottom because gano will release a lot of spore. So we don't want the spore to mess with our oyster lah. The good practice is that to use different different fruiting chamber to grow different mushroom. But we only have one, so yeah, we do what we have. We use what we have. Okay, so let's see closely. So that's how we put our block in our in our chamber, fruiting chamber. We call this fruiting chamber. Okay, so this also. Okay. Okay, so there. Okay, what else? Okay, we use this this growth tent is from AC Infinity. The reason why we prefer this brand is because you can control it easily, and also you can control it through phone. So let's say let's me take my phone. Okay, so here there is some light there. So okay, also there's some light there, reflection. Okay, so here, so we have fruiting chamber there. You have incubator over there. We have incubator one, the one. So you can basically uh, control our chamber using our phone. For example, let's say I want to sort of like to turn off this light there. So I just go to our fruiting chamber here, and then this is, you see, I don't know it's sharp or not. Okay, this is the light here, the light there. So we put our light to the schedule, meaning that we set our light to on and off every 12 hours lah. So to mimic our the natural daylight, 12 hours light and 12 hours dark. But you can also override this thing. For example, let me show you how to sort of like turn off this light. So let me put here, so you can see there off. So I click off there, so it will turn off this thing. Okay, but because we set this thing to schedule, so where is schedule? So the schedule. Okay, so the schedule, so meaning that in 48 minutes this thing will sort of like turn off. Okay, so that's the beauty of having this controller lah. So you don't need to manually come and turn off. And also our humidifier here, we set it to what we call at 90% humidity. And also for this, what we call, for this aircon, we use our what I call our external portable aircon there. So over there, I don't know whether you can see or not, we set our temperature to 25 degrees Celsius. Not sure whether you can see or not, but that is the 25 lah. So when the temperature inside this chamber is above 25, then the aircon will kick in. Now the aircon is kicking in, so that's why you hear the aircon there. Okay, so it will kick the cool air inside. And also we have this what we call over there. I don't know whether you can see there. They're also like exhaust fan lah, so that it can replace the air inside with the new air lah. Because here we have the where can you see? So this thing. So you can see here. This is where the air coming in. Because the mushroom, there are few things that's very important to grow mushroom. The most important part is humidity. You need to humid environment. The second thing is fresh air exchange, or FAE. So we have back there. Here, this is our CO2, what we call CO2 sensor. So we have sensor CO2 inside that. So here is 553 ppm lah, which is okay lah for mushroom growing lah. You need below 500 like that. 500, 600 is okay. For mycelium, it's okay if you have high CO2, but for mushroom fruiting, you need low CO2 inside the chamber. So here, where is the CO2? Where is the CO2? I think here. So this is our CO2 sensor here. Okay, our CO2 sensor. And this is our what we call? This is our temperature and humidity sensor. And what else? And here we don't have what we call the fan, because not really needed actually. I mean, you can have it, it's nice, but because we have what we call this air being exchanged in 1 hour, we exchange it 4 times. So I think this air inside this is always being replaced lah. So we maintain our good FAE, fresh air exchange, inside this chamber. Okay, so let's look our mushroom there. Okay. So this already quite mature lah. Normally when you want to harvest, you harvest it early on lah. Maybe like 6 hours before this recording is much better lah. There are no wrinkle, there are no curly thing. But now it's already late. But it's okay. This is just for test. But this, if you wait another 12 hours, it's already big and nice lah to harvest. So I think that's our fruiting chamber, where we fruit our what we call our mycelium. Okay, so next here is basically our test substrate. We have many substrate there. We put just on the side of what we call our fruiting chamber, so that's easy to see and organize. Normally when you buy this thing online, typically they are made of sawdust. Okay, so here we have also example of typical substrate that is used for mushroom. So normally in Asia they use this what I call rubber tree sawdust. So let's see here. So this is rubber tree sawdust. Rubber tree sawdust. Okay, rubber tree sawdust from rubber tree. Rubber tree is hardwood, so they have a lot of cellulose inside. That's why it's good for what I call for mushroom to grow on that. Okay, so this is rubber tree, and typically they mix it with what I call with rice bran lah, or in Malay we call it dedak. Okay, so this rice bran, example of rice bran. This rice bran, and also they mix also with the kapur. Kapur in Malay, in English is basically lime lah. This is calcium carbonate. Calcium carbonate is just lime lah. Okay. Typically in industry they mix this sawdust as a main substrate. They mix with the rice bran. Rice bran is basically a protein source to what we call to make the mushroom more bigger like that. And this also mixed with this lime just as a buffer, pH buffer. So normally they mix with the ratio 100, 10 and 1. So 100 part of sawdust add with 10 part of rice bran, add 1 part of lime. So 100 kilo, 10 kilo, and then 1 kilo of that you mix together, and then you put into the bag here, and then you inoculate with the spawn, and then you get this thing. So that's how industry did lah in Asia, at least lah. They use sawdust. So this humidifier, before I forget, this humidifier also you need to top up. You need to not to forget to top up. So we just use tap water. We don't use any distilled water or whatever, because yeah, I mean tap water is sufficient. So we use tap water. When it goes down here, we just what I call top up it. So this normally it will last around 2 days like that lah. So you need to top up every 2 days. And because this chamber is fully enclosed, this our what I call the door. So the humidity is trapped inside. So that's good lah, because you maintain the humidity within this thing. You can see the bottom here, there are some water there. Okay, some water. So meaning that this thing sort of like maintain, they don't escape much lah. Of course this thing will escape, because if you look here, I don't know if you can see or not here, there some hole there to allow some fresh air exchange. The humid air, the mist can go here lah. But because we set our exhaust fan not to be on every time, meaning that we set it so that it will on in 1 hour, it will on 10 minutes for 4 times only. So sometimes this humidity inside this chamber will become stagnant, and that's the reason why you can see sort of like some water here, some condensed water here, which is good lah. At least it will maintain the humidity inside this chamber, because humidity is one of the most important thing in growing mushroom. Oh, by the way, you can see here, this is cacat. This is basically, I don't know, the mushroom they grow inside. Anyway, they don't want to grow outside there. Anyway, it doesn't matter. Okay, so this is our fruiting chamber. Okay. One more thing about this control unit is that you can basically trace the what you call the, where is the thing, this thing, right? You can basically trace the what happened to this chamber in the real time lah. So you know if something happened to your the red one is basically temperature. So you can sort of, I mean, let me make it smaller like that. So you can see, you can trace whether something happened to your chamber, is there any spike in temperature and so on. So it will record everything. This thing will record everything. So it's good for you lah, because you don't want to be looking at this chamber every time maybe once in 2 days you check, but not every time. But if you see something wrong, then you can always check the graph, what happened in terms of humidity, in terms of what we call the temperature and so on, so you can troubleshoot later on. One more thing about this growing the mushroom, actually you don't really need this fancy, what we call fancy fruiting chamber. You can just use this box. If let's say you are start to grow, you can just buy this. This we bought it online. You can just buy it online. They sell it online. This is TKV, is basically tiram kelabu variant, basically grey oyster mushroom lah. So they sell it for about RM2 or RM3 per block, less than $1 lah, 50 cent I think, 50 cent like that in USD. So you can just put it outside, and even if you put it outside, you can see the mushroom still grow, because oyster mushroom is high temperate mushroom, meaning that it can grow even though the temperature, they have like more, they have like tolerance to temperature fluctuation lah. So even though the temperature is like 30 degree, still okay. So we just put this, basically we just put outside in our lab, and it just still grow, but this time it's so stubborn, it grow at the back. Normally we open this thing to trigger the grow. We open this stuff, that's why you see all our block there is open. So we open that, actually we open this thing, so we open that thing so that the humid air can meet the mycelium. For this block, for this normal block, normal plastic bag like this, we just sort of like make an X. We take a scissors, not scissors, the knife, and we scratch, we tear it apart, and then it will expose the mycelium to this humid air, and then it will grow lah, like this stuff, like this ganoderma there. Okay, like that ganoderma there. Okay, so that's why I said you don't need this fancy stuff, but it's good in order to maintain everything in one place. But if you don't have this, what we call this fruiting chamber, you can always buy the block or make the block yourself and put it outside, and let it see. Okay, let's see whether it's grow or not. Or you can put inside the, what we call, inside this box. Normally this in order to retain, what we call, the humidity lah, you put your block and then you put some wet clothes on top of that in order to mimic the humid environment. And then to prevent the light, you can just put like this, close like this, but don't close fully lah, because when you close like this fully, then there are no air exchange, so that's will cause the mushroom become the stem become long like that. So that's the sign of not enough air exchange lah. So this is the old one, so you can see that is the spore. So this is the old one, so that's why it's already just like that. It's like more than like 3 weeks already, so we just put it like that. Anyway, it's grown nice before, but because mushroom, if you don't harvest it in 2 days, then it just become like that, spoil, soggy, and yeah. Just I want to show you that is the spore, that is spore of the mushroom. Okay, so you can just use this simple box in order to grow your first mushroom. For Ganoderma release a lot of spore. So this is our Ganoderma. We just put in the box, and then we just left it like this, open a little bit like this. You can see all this yellow stuff here is a spore of Ganoderma here. This spore of Ganoderma. Can you see there? Yeah, that's spore. Oh, that's all the spore. So basically, you can sprinkle this on the agar, and then you get the mycelium, and then use that mycelium to make a spawn, and then you put again, and then you get the Ganoderma again lah, fruit mushroom again. So that's the life cycle of Ganoderma. So here you can see we just put here without any fancy environment, fancy equipment. We just put in the box. We left it like some air to get in and get out, and it still grow. Yeah, some mushroom is very tolerant lah. For example, Ganoderma and this oyster mushroom is very highly tolerant to fluctuation of temperature, humidity, and so on. So yeah, that's basically it lah. So this is how we grow mushroom in our lab. Bye.